I'm Jinho from Salk Institute, Songan Laboratory. Breathing is unique. If I say this, you may ask why. Let me tell you why. Think about these situations. Now you are swimming, so your head goes above or below water. Now you are drinking because you feel thirsty. Now you are speaking to someone, like what I'm doing now, or you are singing. In all these behaviors that require orofacial activities and the airway control, you are now controlling cycle specific. Even at the single cycle level, you are synchronizing your breathing to your behavior. That is very unique. You don't synchronize your heart rate or gastric movement to your singing or speaking. So we call this as breathing behavior entrainment. In other words, behavior control of breathing. There's one more reason. So can you consciously control any of these physiological features? Like, do you control your heart rate? But controlling breathing is very easy. It's this easy. We call this volitional control of breathing. So these are some features of breathing that stand out from all the other physiological features. And importantly, if you check out some literature written by famous people in this field, they say the neural brain circuits for these features, it's unknown. So that's what I've been studying. So to find out such a circuit, the first step is to identify the top-down breathing circuit. To find out that, I made some criteria. First, I thought this kind of mediator, behavior and breathing connection, might be a downstream of the prefrontal cortex. And this should be upstream of the very breathing centers in the deep down medullary or pontine regions. And I was also more interested in the inhibitory control because I thought in many biological disorders, lack of inhibition is the cause of many diseases like epileptic seizure because of lack of GABA, something like that. So by searching through Allen Brain Atlas, a lot of database in the Allen Brain Atlas and some tracing data from our own lab, I narrowed down some candidates that satisfy these criteria, and I found one. This is one of the candidates, pontine reticular formation caudalis. First, to see if this region actually receives the input from the prefrontal cortex, I injected the CTB retrograde tracers into the PNC. The reason I have another color here is because I realized that this dorsal region also receives the prefrontal input. So if there is any spillover from here to the dorsal, then the tracing result changes. So I injected two different colors. And this is the result. I basically found two dissociated different subsets of neurons in the prefrontal cortex, one in the dorsal anterior cingulate cortex and the secondary motor area. Another was in the prelimbic cortex and the anterior cingulate ventral area, like this. Then the question is, so I found two different types of neurons. Are they really controlling breathing rhythms? To answer that question, I expressed the channel rhodopsin into the prefrontal cortex with the injection of AAV retrovirus into the PNC or DTN area. And as I activate these neurons using blue light, I was able to induce slower breathing with reduced amplitude as well. Conversely, if I activate the ventral side of the PFC, which project to the DTN, it causes faster breathing. So answer to the first question is yes, we propose as above. We basically found two distinct subsets of neurons, one for slow breathing and another for fast breathing. Then the next part is, if that's the function of these neurons, then I must be able to observe some neuronal activity patterns that's actually correlated with the breathing patterns in certain behaviors. So I expressed the calcium activity marker GCAM7 as shown here using the AAV strategy. And I also simultaneously recorded breathing patterns using this thermistor methodology. And this is an example trace that I recorded from just a home cage. Nothing was given. This is just voluntary change. Interestingly, what I found was that when there is fast to slow transition of breathing rate, you can see that there is a transient increased peak of the calcium activity of ACC-PNC neurons, the slow breathing neurons. Conversely, if I align the breathing patterns, breathing rate to the occurrence of the peak, I was able to see the prediction of the drop of the breathing rate. Interestingly, in more dramatic cases, I was able to see some breath holding. sometimes mice hold their breath like something like that. And that was also correlated with the chiasm increase. Behaviorally, when I look into the video, this was correlated with some locomotor changes like turning their head direction because of movement changes. But is that all? Is this prefrontal circuit just for some locomotion or posture related breathing changes? And my answer was no, it must be more general. So I tested with some specific behaviors that might require some breathing patterns. One, swimming. So let mice swim in water, especially during the initial phase of swimming. This is pretty well known. I was able to see apnea or breath holding or some delay, slow breathing, those kind of variations in the breathing rhythms. And I was also able to see the huge increase of chiasm activity during that phase of swimming. I found one thing very interesting. So this is well-known phenomenon called diving reflex. They say when we suddenly touch the water with our nasal area or facial area, there is an immediate reflex-like changes in breathing or physiological features after touching the water. But I did this frame-by-frame analysis to see when exactly the mouse touched the water. And interestingly, I found that the increase of chiasm activity occurs before touching the water. It's before. So my interpretation is that in these kind of cases, like when we're able to see what's going to happen, like we're predicting that we are entering water, then maybe these prefrontal neurons are preparing to change the breathing. Next, second behavior is drinking. This one was relatively simpler. So I just gave mice some droplets of water when they are thirsty and see when exactly the mouse drinks it. When it drinks it, there is a breathing change and chiasm increase. So I was able to see this average trace. Next behavior, phonation, generating vocal sound. We can generate our vocal sound only during exhale. We cannot generate sound during inhalation cycles, so it requires breathing control. Well, it could be more interesting if I can study some singing behavior in mice, but our lab mice are not able to sing. So I chose a different form of phonation behavior as squeaking. So when I gave mice some food shock, I was able to observe some sound patterns like chick, 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 chick. This is exactly what mice generated. And that was synchronized with the breathing changes, as you can see. And when I did that with the chiasm imaging at the same time, I was able to see that the synchronized patterns of all three, sound, breathing, and the chiasm signal of the ACS-PNC neurons. So this is a summary of the second part. In various situations, I was able to see the activation. But you may ask, where is the ACVDT and the fast breathing data? I'm doing this experiment from this week. Part 3, breathe slowly and experience tranquility. Breathing is not only important for some acute changes of behavior, but also for long-term tendency of behavior, such as the behaviors related to anxiety and depression. To see if there is such a tendency in mice as well, I did this experiment with thermistor implanted breathing recording or thermistor and chiasm recording at the same time. I observed that the mice that are staying longer in the light zone, which is an anxiogenic environment, they were having slower breathing rate. So interestingly, I found a correlation, like the breathing pattern and the level of anxiety was correlated in mice. And now I'm adding more animals in the chiasm imaging group to see if how the chiasm pattern can change the breathing patterns in the light zone. Then I saw some correlation. How about
just over 9 minutes?
Yep, thank you. How about the causation? So I tested with some general listening experiments to see if I can change the behavior. This is some control experiments. Even though I activated the ACS-PNC slow breathing neurons, there was no change in RTPP, the emotional behavior. But interestingly, if I activate the same neurons in the presence of a threat, it can change, it can abolish the fear behavior. Or the activation of anxiolytic in an EPM test. I'm going to do the light-dark preference test as well. So the summary is like, I saw a correlation between slow breathing and the anxiety-related behavior. But when I activated the slow breathing neurons, there was no change in RTPP, but this could block the fear responses. these are some directions that I'm pursuing in the current study, um, including something about the connections, and these are shown in my presentation, which needs some more elaboration. And in the future study, it might be good if we can find some specific cell markers to target um, these neurons with new drugs. And it might be also interesting if someone studies in this circuit in the human brain. Thanks for listening. Do you have any questions?
It was great. Thank you. Any questions from anyone? Let's see. There's one question, a couple of questions here. Let me read them out here. So first of all, beautiful work. How do you know that mice can consciously control their breathing? How could you know? Can you train mice to stop or start breathing when they hear one tone but not another?
So I'm actually looking at the question by myself. That's a very interesting question. Um, let's see. So that's so-called the volitional breathing. We humans are able to do that, but according to the literature, there's no model for mice or any animals, no animal models about the volitional breathing. So actually, in my paper, I'm going to include those behavior components of breathing, but for volitional part, maybe we should develop a new model, like voluntarily for some reward. If mice or rats can control their breathing rate for the reward, maybe that could be one of indirect evidence. But we don't know whether that's controlled by your thought in mice or rats. And can you